Hello, welcome to Notable Nobels, a podcast about the Nobel Prize in Physiology or Medicine. My name is Harrison Doolin. I'm a postdoctoral scientist at the Fred Hutch Cancer Center in Seattle, and I will be your host for this web series. The purpose of this series is to trace key advancements made in the biological and medical sciences, and we're using the Nobel Prize in Physiology or Medicine as a guide. Now, every career has its highest prize, and for scientists, that prize is the Nobel Prize. It's the most prestigious award a scientist can receive, and it marks discoveries that have made a profound impact on our understanding of biology and our ability to treat diseases. Today, we will be examining the 1997 prize in Physiology or Medicine, which was awarded to Stanley B. Prusiner. The Nobel Assembly at the Karolinska Institute chose to give Prusiner the award, quote, for his discoveries of prions, a new biological principle of infection, unquote. We'll be going over how Prusiner was able to prove that a protein by itself was capable of being an infectious disease, and we'll be going over the fascinating mechanism behind how these protein diseases replicate and spread, and we'll cover some recent outbreaks of prion diseases. But first, a little bit of background on Stanley Prusiner. Prusiner was born in 1942 in Des Moines, Iowa. His father was the son of Jewish immigrants from Russia who had moved to Iowa in the late 1800s. His mother's family was from Cincinnati, Ohio, and shortly after Stanley Prusiner was born, he and his mother moved in with his mother's family while his father served in the U.S. Navy during World War II. After the war, his family moved permanently to Cincinnati, where his father built a successful career for himself as an architect. Stanley finished high school in Cincinnati before heading off to the University of Pennsylvania for college. He majored in chemistry, and in the summer of his junior year, he got his first taste of scientific research when he started a research project on hypothermia. He liked the project so much that he continued it into his senior year, and after graduating, he decided to stay at Penn for medical school. In 1968, while getting towards the end of medical school, he was offered a research position at the U.S. National Institutes of Health, or NIH, in Bethesda, Maryland. He spent 3 years doing biochemical research at the NIH, during which time he was able to hone his research skills and learn a great deal about the scientific process. After finishing his term at the NIH in 1972, Prusiner went off to complete his residency training in neurology at the University of California, San Francisco. 2 years later, he accepted an offer to be an assistant professor at UCSF, and he began setting up his own lab. It was at UCSF that Prusiner would do his Nobel Prize-winning research. But before we get into Prusiner's work, let's take a second to recap the research from our previous episode and see how it came to involve Prusiner. You'll remember that last episode, we talked about the Kuru neurodegenerative disease. Kuru was the first prion disease identified in humans thanks to the work of Carleton Gajdusek. Gajdusek had demonstrated in the mid-1960s that Kuru was an infectious disease, and he did that by transmitting the disease to chimpanzees. But he quickly realized that the Kuru infectious agent was highly unusual. Even though the Kuru agent was small like a virus, it produced none of the usual signs of a virus infection. The agent also seemed to take an unusually long time to produce disease symptoms, and the molecular nature of the agent was a mystery. Additionally, the agent seemed resistant to treatments that would inactivate nucleic acids, which was extremely puzzling. Gajdusek thought the Kuru agent was some kind of slow virus infection, and after demonstrating the transmissibility of Kuru, he went looking to see if there might be more of these slow virus diseases out there, and there were. These new diseases were given the name transmissible spongiform encephalopathies. So, to understand what transmissible spongiform encephalopathies are, let's go ahead and define each of those words individually. Transmissible means that the disease can be passed from one individual to another, or in other words, the diseases are contagious. Encephalopathies is a compound word that is made by combining the Greek words encephalon and pathos. Encephalon is the Greek word for brain, and pathos in this context is the Greek word for suffering or disease. So encephalopathies are brain diseases. The word spongiform here is a reference to the microscopic appearance of the diseased brains. When brains from individuals who died from transmissible spongiform encephalopathies were examined under a microscope, scientists saw what looked like tiny holes in the brain, which were actually vacuoles in the neurons. These vacuoles made the brain tissue look like a sponge, so the term spongiform was used to describe the appearance of the diseased brains under the microscope. So, putting all that together, transmissible spongiform encephalopathies are contagious diseases of the brain that usually give rise to microscopic sponge-like appearance of the diseased brains. The three best-known transmissible spongiform encephalopathies in the early 1970s were Kuru, Creutzfeldt-Jakob disease, and Scrapie. All three of these diseases would play a crucial role in the early science of prion diseases. We talked about Kuru last time, so let's talk a bit about Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease, or CJD, was first described in the 1920s by a pair of German neurologists for whom the disease is named. The progression of the disease has a lot in common with Kuru. Like Kuru, CJD patients will have problems with speech and walking. They will also show signs of dementia, including memory loss and personality changes. Also like Kuru, the disease is fatal, usually within 6 to 12 months after the onset of symptoms. Unlike Kuru, which only occurred in Papua New Guinea, CJD is found all over the world, though it is extremely rare. Now, Gajdusek was interested in the possibility that Kuru and CJD might have a similar underlying cause, so in 1968, he decided to try transmitting CJD to chimpanzees, just like he had with Kuru. The result was that the chimps developed the disease, and microscopic imaging of the brains showed the characteristic spongiform appearance. So CJD was the second transmissible spongiform encephalopathy identified in humans. Now, one other disease will be important for our discussion today, and it's called Scrapie. Scrapie was actually the first transmissible spongiform encephalopathy identified, though it got much less attention at the time because it is not a human disease. Scrapie is a disease of sheep and goats. Descriptions of Scrapie can be found as far back as the mid-1700s in continental Europe, but it wasn't until the mid-1800s that scientific study of Scrapie really got going. Like Kuru and CJD, sheep that get Scrapie will have neurological symptoms that include altered behavior and trouble walking. One noticeable feature is that the animals will excessively rub and scrape themselves against trees and posts, like they have an itch that won't go away. And this is how Scrapie got its name, this scraping behavior. Also like Kuru and CJD, death occurs around 6 months after the onset of symptoms. Scientists in the late 1800s floated the idea that Scrapie might be caused by an infectious agent. A team did try to test the idea by inoculating sheep with infected tissue, but when they didn't observe any Scrapie symptoms after a couple of months, they concluded that Scrapie wasn't an infectious disease. However, the epidemiological data of the disease strongly supported the idea that Scrapie was in fact an infectious disease, so the hypothesis persisted. Then in 1936, 2 French scientists were successfully able to experimentally transmit Scrapie from diseased sheep to healthy sheep. Now, the time between exposure to the Scrapie agent and onset of disease symptoms was between 1 and 2 years, giving rise to the belief that Scrapie was some kind of slow-acting virus. This long incubation time between exposure to diseased tissue and onset of symptoms was just what Gajdusek had observed with Kuru and CJD. It's at this point in the story that we can bring in Stanley Prusiner, our Nobel laureate. In 1972, when Prusiner was just starting his residency at UCSF, a woman with CJD arrived at his hospital. This was the first time Prusiner had heard about the disease, and here's his description of what happened next. Quote, I admitted a female patient who was exhibiting progressive loss of memory and difficulty performing some routine tasks. I was surprised to learn that she was dying of a slow virus infection called Creutzfeldt-Jacobs disease, CJD, which evoked no response from the body's defenses. Next, I learned that scientists were unsure if a virus was really the cause of CJD, since the causative infectious agent had some unusual properties. The amazing properties of the presumed causative slow virus captivated my imagination, and I began to think that defining the molecular structure of this elusive agent might be a wonderful research project. The more I read about CJD and the seemingly related diseases, Kuru of the Fore people of New Guinea and Scrapie of sheep, the more captivated I became. Over the next 2 years, I completed an abbreviated residency while reading every paper that I could find about slow virus diseases. In time, I developed a passion for working on these disorders." Unquote. Well, Prusiner was given the chance to explore his scientific passion when he was appointed a professor at UCSF and set up his own lab. He immediately realized that the task he had given himself wasn't an easy one. One of the most challenging aspects of working with the transmissible spongiform diseases was the extremely long incubation times required for disease symptoms to occur. Also, while Gajdusek had made excellent progress studying the diseases in chimpanzees, apes were not an ideal animal to work with. So chimpanzees require large, highly specialized facilities, and also, since 1990, they have been listed as an endangered species, and because of this, no labs nowadays use chimpanzees as lab animals. Prusiner could have tried working with sheep and Scrapie, but again there was the problem of the long incubation time and large facility needed to house the animals. Prusiner, who was just getting his lab started, wanted to work with a small animal model, and ideally one that could produce disease symptoms relatively quickly. After some initial experiments, he found that the Scrapie agent could replicate and produce disease symptoms in hamsters after only a few months. This was much faster than working with Kuru or CJD, and hamsters were small and easy to work with. Once he had his Scrapie hamster model system in place, Prusiner began his experiments to try to figure out the identity of the Scrapie agent. Like Gajdusek, Prusiner thought Scrapie was caused by some kind of novel virus, but after performing numerous experiments to purify the Scrapie agent, the data consistently suggested that Scrapie was caused by a protein agent that did not contain any nucleic acids. Now, this was puzzling, since nucleic acids are the basis for how every living thing on the planet replicates. The idea that a protein in and of itself could be an infectious agent was revolutionary. While Prusiner was originally skeptical of the idea, his own data continued to insist that the Scrapie agent was likely a protein and did not contain nucleic acids. In 1982, he decided to publish his findings in the journal Science. He suggested in the paper that the Scrapie agent be used to define a new type of infectious agent that was protein-based, not nucleic acid-based. He gave this new class of infectious agents the name prions, which was a hybrid word derived from the phrase proteinaceous infectious particle. The response to Prusiner's paper was rather extreme, and not in a good way. Here's how he describes the blowback he received. Quote, "Publication of this manuscript, in which I introduced the term prion, set off a firestorm. Virologists were generally incredulous, and some investigators working on Scrapie and CJD were irate. At times, the press became involved, since the media provided the naysayers with the means to vent their frustration at not being able to find the cherished nucleic acid that they were so sure must exist. Since the press was usually unable to understand the scientific arguments, and they are usually keen to write about any controversy, the personal attacks of the naysayers at times became very vicious." Unquote. Prusiner points out that the easiest way for his opponents to settle the controversy would be to find the presumed nucleic acid of the supposed scrapie virus, but nobody found it, including Prusiner himself, who regularly pointed out that he and his colleagues had tried harder than anyone to find the nucleic acid to no avail. So to shut down the skeptics once and for all, Prusiner put all his efforts into characterizing the scrapie protein prion. One of the questions Prusiner had to answer was, where does the scrapie prion protein come from? All living things make proteins by coding those proteins in the form of genes. The genes contain the information for making the protein, and the genes are stored in the form of a nucleic acid molecule like DNA. But Prusiner had said that prions don't have nucleic acids. So where was the gene that coded for the prion protein if it wasn't part of the infectious particle? Prusiner and his colleagues published the answer to that question in the journal Cell in 1985. They had worked out the amino acid sequence of the scrapie prion and then worked backwards to identify possible DNA sequences for the protein. They then checked the genome of hamsters, again their animal model for scrapie, to see if there was a gene that coded for the prion protein in the hamster genome. And they found the prion protein, also called PRP, was in fact coded for by a gene in the DNA of the hamster, and not just hamsters. Prusiner's team also checked the genomes of mice and humans and found the gene for PRP there as well. Eventually, it became clear that the gene for the prion protein is found in every mammalian genome. This was an important discovery as it explained where the prion protein came from. It came from, well, us. But it also generated even more questions. If every single mammal has a gene for prion protein, why aren't we all dropping over dead from neurodegenerative diseases? The answer to this question was published in the journal Nature in 1994, and it was one of the most sensational discoveries of infectious disease biology. Now, to understand how scrapie and other prion diseases work, we first need to go over a bit of molecular biology. As stated earlier, cells contain genes that contain the information for making proteins. Now, proteins are some of the most dynamic and diverse molecules in nature. They are also the main workhorse molecules of cells. In other words, proteins enable cells to do things. Now, proteins are molecules and they have three-dimensional shape. They occupy 3D space, and their 3D shape will help determine their role in the cell. This is usually summed up with the phrase, form facilitates function. Think of the silverware that you use at a dinner table. The knife, fork, and spoon are all made of the same metal, but that metal can be molded and shaped into different forms that can do different things, like cut bread or hold soup. Proteins are similar. A protein can be twisted and shaped to occupy a specific structure or shape in three-dimensional space, and the structure of the protein will determine the kinds of things that protein can do. Now, the prion PRP protein can have two different shapes. This was a crucial finding. In almost everyone on the planet, the PRP protein exists in a harmless shape called PRPC. However, under certain conditions, that PRP protein can change shape, and this different version of the PRP protein is called PRPSC, SC for scrapie. This SC form of the PRP is the disease form of PRP. Individuals suffering from prion diseases have extremely high amounts of the PRPSC protein, while healthy people do not. Scientists working in experimental models of scrapie were able to show that after infecting an animal with scrapie, levels of PRPSC increased over time, and that PRPC seemed to be converting to the PRPSC form. This led scientists to wonder what was causing the conversion of healthy PRPC to disease PRPSC. The astonishing finding that was published in the 1994 Nature paper was that PRPSC, the disease version of the protein, that was the catalyst for converting the normal version of the PRP protein into the disease version. So prion proteins replicate by converting the normal PRP proteins that are present in every single mammal, including us, into disease prion proteins. So a good analogy here is to think of the stories of werewolves or vampires, like the older legends of these creatures. These monsters will bite humans and convert them into monsters like themselves. PRPSC is just like that. It's a monster molecule that grabs hold of harmless PRP molecules and converts them into disease-causing prions. Those new prions can then go on and do the same reaction, converting harmless PRP into disease prions. This remarkable finding finally explained how a protein by itself could be an infectious agent, and it revolutionized both infectious disease biology and molecular biology. In recognition of his contributions to the discovery of prions, the Nobel Assembly at the Karolinska Institute awarded Stanley Prusiner the Nobel Prize in Physiology or Medicine in 1997. Now, you might think that prion disease occurs because the misfolding of the PRP protein into its pathogenic form results in a loss of function of the PRP protein. In other words, one might imagine that normal PRP protein performs some important and necessary cell function, a function that it can't do when it's converted into its pathogenic form, right? Form facilitates function. So you might hypothesize that it's the PRP loss of function that makes the neurons sick and causes disease in these transmissible spongiform encephalopathies. However, that turned out to be incorrect. Scientists have deleted PRP from the genomes of mice, thus creating a loss of function, but the mice don't develop prion disease symptoms. Even more strangely, the mice don't develop any symptoms at all. That's right, mice with PRP grow up and reproduce and eat and drink and sleep and appear mostly normal. So scientists still don't really know what PRP does and why it's in our genome. And we still don't really know how misfolded PRP protein causes neurodegenerative disease. However, it is absolutely clear that the PRP protein is required for disease to occur. Mice lacking PRP do not develop disease when infected with prions, so PRP protein is necessary for the disease. But there's still a lot of work left to be done to better understand how prion diseases work. So, with that being said, what is the state of prion diseases in the world today? So what are your chances of getting a prion disease? Well, there are 3 different ways you can get a prion disease. The first way is spontaneously. Now, spontaneous here does not mean a person's prion disease didn't have a cause. It did, it just means we don't know what the cause was. About 85% of prion cases in humans are a form of CJD called spontaneous CJD. So these people, they have no known predispositions to CJD and no known contact with infectious prion proteins. It's likely that through some unknown mechanism, something triggered the initial misfolding of PRP protein in these people, which over time led to self-amplification of PRPSC and eventually to disease. So these cases are almost always in older adults, and there's about 5,000 spontaneous CJD cases each year worldwide. The second way you can get a prion disease is through genetic predisposition, or basically inheriting one. A small number of people have an inherited form of CJD, and they make up about 10 to 15% of human prion cases. These people have mutations in the gene for the PRP protein, and these mutations greatly increase the odds of PRP converting into its SC form. These people usually developed CJD at a much younger age compared to people who get spontaneous CJD. The third and most notorious way you can get a prion disease is through exposure to infectious prion protein. Now, depending on the year, these cases make up between 0% and 6% of prion disease cases. So to wrap things up today, let's go over how you can acquire a prion disease. Specifically, let's talk about prion diseases in cattle and deer and the United Kingdom mad cow disease outbreak. In 1984, a cow in the UK county of Sussex died after displaying unusual neurological symptoms. The next year, more cows from the same herd also died after showing similar symptoms. Soon, cows from herds all over the UK were dying of this mysterious illness, which was given the name bovine spongiform encephalopathy, or BSE, a new form of transmissible spongiform encephalopathy. In general terminology, BSE is known as mad cow disease. By the early 1990s, tens of thousands of cows in the UK were dying of the disease each year. The source of the problem was soon realized to be contaminated animal feed given to the cows. In order to increase protein content in milk, cows in the UK were being given a kind of animal feed called meat and bone meal, which was made from discarded animal tissue. It's very likely this feed contained contaminated prion proteins that triggered the outbreak in the cattle. To stop the spread of the disease, the UK banned the use of the feed on cattle farms. But while this epidemic was going on, meat and dairy products from cows were still being sold in the UK, as it was generally considered that the disease would not pose a problem to humans. After all, the UK had been through many outbreaks of scrapie in sheep, which had never been a problem in humans. However, it became tragically clear that this assumption about the risk of BSE to humans was wrong. Starting in 1995, there was an uptick in deaths from CJD in the UK. The first death was a 19-year-old with no known family history of CJD, so that was a big alarm bell that something was amiss. Remember, CJD is mostly in older adults. Over the next year, more cases of CJD occurred in the UK, including 10 cases in people under the age of 42. This led the UK government to conclude in March of 1996 that these cases were in fact linked to the outbreak of mad cow disease in cattle, and that these people likely acquired the disease after coming into contact with diseased meat. In response to this announcement, the European Union, which the UK was still a part of at the time, issued an international ban on British beef exports. The announcement also led to a great deal of panic in the general population, since CJD is, to this day, a 100% fatal disease with no known cure. Celebrities like Oprah Winfrey declared that they'd stopped eating burgers, and farmers in both the UK and around the world suffered billions of dollars in losses. Eventually, the epidemic was brought under control, but not before an estimated 176 people in the UK died because of mad cow disease. While mad cow disease has been largely eradicated and there is no known risk to the food supply nowadays, the outbreak highlighted the risks of animal-acquired prion diseases and led people to wonder if there are other sources of animal prion disease that we should worry about. Now today, there is a great deal of interest in a prion disease in deer and elk that leads to a disease called chronic wasting disease in these animals. Now, it's still an open question if chronic wasting disease can be transmitted from deer or elk to humans, though health agencies emphasize there does not appear to be any risk to the public at this time. However, the disease has been spreading steadily in the deer population in the US and Canada, and it seems to be making its way into new territories. So, for the moment, health officials and scientists are continuing to monitor the spread of the disease, and it is recommended, out of an abundance of caution, that you do not eat meat from a deer or elk if it is suspected that the animal died of chronic wasting disease. There is still, honestly, a lot of work to be done with understanding prion diseases and in developing ways to cure or prevent them, and it will be fascinating to see what breakthroughs are made in the years to come. Okay, that concludes this episode of Notable Nobels. This episode was recorded on June 14th, 2025. I want to thank Digital Mind Productions for providing the music and Nicholas Wynn for the graphic design. This episode also marks the conclusion of Season 1 of this podcast. Woo! It took me a lot longer than I thought it would to get to this point, but here we are, we made it. Woo! Now for Season 1, we focused on Nobel Prizes in physiology or medicine related to infectious diseases. Season 2 will focus on the closely related topic of Nobel Prizes in physiology or medicine related to immunology. But before we do the first episode of Season 2, I will be releasing a special episode that you'll definitely want to check out, so keep an eye out for it. Thanks again so much for listening. We'll see you then.